American Gene Technologies has been in gene and cell therapy now for a little over 10 years. We adopted a platform from NIH that was being developed by Roscoe Brady, who's well known for having cured Gaucher's disease, and was a pioneer in enzyme replacement. His drug was out-licensed to Genzyme and was selling $1 billion a year, and he went back to lab to look at other ways to fix metabolic diseases, and he was focusing on a lentiviral vector platform. So we had adopted that in 2008 when he was closing down his lab. And the approach that we took, given my background in the computer industry, was to look at the software for the human computer in the same way that you look at software for the digital computer, that it's a layered model, and that the important thing was to know the fundamentals in software rather than being really fixated on a particular viral vector platform. So we're vector agnostic, and we work with everything in the lab. AAV, integrating lentiviral vector. We've developed a non-integrating lentiviral vector as an innovation in the industry. We've done 2,000 or 3,000 constructs that have taught us what works and what doesn't work in terms of cargos for any viral vector system. We work with replicating viruses, experiment with CRISPR, and the point being that it's all about creating value and solutions for the patient. That's the critical component, and expertise across all of the elements of getting the proper genes to express at therapeutic levels in a safe way and a targeted way are really the three legs of the gene and cell therapy stool. So this has been very fruitful for us. We've driven down the cost of developing and the time in developing viable drug candidates, and we've done about 15 different proofs of principle, and we selected 3 of them as we started to move into clinical development. Those 3 products are an HIV cure, a phenylketonuria cure, and an immuno-oncology asset, which is the stimulation of gamma delta T cells for the therapy of epithelial solid tumors. So I'm going to take those in reverse order. First, I'm going to cover the immuno-oncology just because that's so hot. There's a lot of approaches to immuno-oncology, and we think that there's sort of a range of efficacy and a range of safety that they all fall on, but the ideas that we have really gotten behind is by utilizing natural cells in the body instead of trying to modify cells in the body in order to create an immuno effect. Instead of firing up a CAR T or modifying the immune system in order to get it to chase cancer more effectively. The best way to do it is to use the cells that are already surveilling for the type of cancer that you're interested in, and to modify the cancer to make it infinitely attractive and stimulatory to the natural immune system. This avoids a lot of the dangers that, and sort of problems that have been illuminated by CAR T. We're big believers in CAR T. We think it's a great product. I'm not denigrating that or diminishing the successes there. But what I believe we've discovered is that you can stimulate the V delta 2 gamma delta T cell population in a patient to attack epithelial solid tumors in a highly effective manner. Now, why is that? This population of T cells is unique in that it has 2 receptors. One is a TCR, a gamma delta chain, and the other one is an NK receptor. The NK receptor is the targeting molecule. That receptor has never been seen in science, research, or in medicine to ever misrecognize. It is exquisitely specific to malignancy. Now, that one subpopulation of gamma delta T cells is 3 to 5% of your circulating T cells. It surveils your entire life long for small epithelial malignancy. And your theory is as good as mine about why occasionally cancer gets ahead of your immune system. I think that accrued stress over a lifetime and maybe a small diminishment in the efficacy of your immune system over time, you hit a critical mass point where your chances go up. So with age, your chances of getting cancer increase. But what is cancer? Cancer is just those small malignancies that escape surveillance. And once they're growing like a balloon and blowing up, of course, the surface area is getting big fast, and then they're ahead of the immune system. Well, what we do is we just take a small amount of a proprietary viral vector. We're using lentivirus, in this case, to carry it, but our construct causes the upregulation of a danger signal that stimulates the TCR on a gamma delta T cell. This is like pressing down on the gas pedal, basically. The TCR does not retarget the T cell. This is very important because if we're going to clear cancer in a solid tumor, we need to get all of it, because even a small amount could go ahead and regrow the cancer. So when we are able to modify 20% of the primary tumor and start stimulating it with it's something called IPP. These patents, by the way, are issued. You can just look them up. But what we do is we're overexpressing this 5-carbon pyrophosphate molecule. That's attracting, training, proliferating, and most of all, activating these gamma delta T cells. They eat at 300 to 600 times their normal rate. So the primary tumors are obliterated. In the mouse models, we've done 1,000 mice on this in a variety of epithelial cancers. You're getting complete remission, zero cancer, in 85% of the mice. And this is with abscopal effect. What I mean by that is that the T cells that are circulating around the host are clearing other epithelial cancers that are untreated in the body. This includes the secondary tumors and metastases of the cancer that you're treating, but it also is getting unrelated epithelial cancer. So we can go ahead and put liver cancer and breast cancer in a mouse, treat the liver cancer, and the breast cancer goes away as well. So this is the results on this are startling. We have a data room on this. We expect to get it into the clinic in 2020. Our collaborator is Stanford Medical School, Dean Felsher. He's a global expert in hepatocellular carcinoma. We think that's a good starting point for this technology to prove it out, to get the proof of concept. And that would allow us to move into other cancers. Of course, liver cancer is an unmet need, high mortality rate, short endpoints. There's no way to cure even a relatively early stage cancer. So let me just go ahead and really quickly. Obviously, we didn't have time for this kind of slide presentation, but this here is what I was telling you about, is by treating just the primary tumor, upper left there, with this immunotox vector. We're just converting a small number of the cells to secrete the peptide. It only stays within the microenvironment of that primary tumor, and this has like a vaccine effect on these gamma delta T cells. They're recruited, that green one, and proliferated and activated, those yellow ones. They obliterate the primary tumor and the metastases. The results are superb. So we've been able to show that in control mice, you have an average lifespan, or let's say that they're all dead by 60 days. By just putting in the vector, you're slowing down the metabolism of the cancer. That's why we're getting a slight life extension there. We don't think that that's important. That's just a result of the fact that the cells can't divide as rapidly while they're doing other work of putting out this molecule. But when you add gamma delta T cells, these mice essentially just live. That line goes out very, very long. We just need to euthanize them to see how it's going. When we frozen section the tumors, we have zero. I mean, absolute zero cancer in them. So these gamma delta T cells proliferate and they infiltrate the tumor, and they're highly, highly effective. That's just a gross picture for you to really set it in concrete with you. So on the left, we have the control group, and on the right, we have the treated group. See the guy who's looking a little bad on the left-hand side there? We think that's from tumor lysase disease. That's why it's 85% complete remission. But we think that the tumors start at about the size of the head of the mouse. So it's a lot of tumor mass. If it breaks down too quickly, you do get a poisoning in the mouse. This shouldn't be a problem in humans. We think this is going to translate beautifully. So while we're waiting around for the cancer cure, we're not sitting on our hands. We're going to get a cure for HIV into the clinic later this year. We are in the latest part of the IND enabling and finalizing the IND that we think will be submitted in the third quarter of this year for an HIV cure. It is a CCR5 knockout, but it has a unique autologous cell therapy which reduces the cost of delivering this substantially and increases the reliability. The problems in CCR5 knockout in the past have been a bone marrow transplant is not a practical way to treat, 1 million patients in the United States. Obviously, it's a very dangerous and expensive operation, but the London patient is encouraging in terms of the overall theory. And so, and Sangamo got some progress there using a zinc-fingered nuclease, but the problem there is that you have to clip both alleles in the same cell in order to get it immune. So we're using a lentiviral delivered micro RNA scaffolded shRNA that knocks out CCR5. So we're getting it reliably on both alleles. We're getting 90% transduction on the T cells. We have an basically an autologous cell therapy that focuses on HIV-specific T cells only. And we're able to get between 10 and 500 million HIV-specific CD4 positive helper T cells per patient in 11-day treatment for less than $30,000 worth of lentiviral vector on an enriched mix. So there's a whole process in there that we've patented that isolates the HIV-specific T cells, stimulates them, gets a very high transduction rate, and gives a massive quantity, which is well above memory effect. So what we're seeing in the blood samples, we've run 9 patients volunteer blood through this, and the results are remarkable. So you have this cell product that you can toss as many infected cells into as you'd like, as much HIV as you want, and it sterilizes itself. Sterilize. In other words, it's getting the viral reservoir as well. So that's really an exciting finding that brought NIAID to us, because if you change the target cell and you change the cargo, this would be effective against a variety of chronic viral infections. So we'll be doing an article with them. We just signed a collaborative research agreement. They're repeating our experiment in their lab. So I think we've got some really good people, not just sort of outside of the company, but if you look at our HIV team, it's really the dream team of HIV. I encourage you to go ahead and look at the site and see of some of our collaborators. We have a data room on this as well. As I said, we'll wrap up the IND in the third quarter. We think we'll treat our first patients in the fourth quarter. We think by the first quarter of next year, we'll have efficacy data. It's a 15-patient open label test. And there's some significant interest from partners on this. So those are our 2 main products. So we have a platform in immuno-oncology, which could go across all the epithelial cancers. We have a platform in immunoregulation for chronic viral infection, which could also go across a number of different chronic viral infections, HTLV, human papillomavirus, hepatitis B, Epstein-Barr, herpes, and so forth. And we have a monogenic program, which I don't even really have time to talk about, but we've just completed testing in mice on our phenylketonuria cure, and we've been able to show that it's within the therapeutic index of our current drug candidate to restore the normal processing of phenylalanine in patients. So we're the first ones to cure this actually in the mouse model with the human gene. And I've completely taken our time, but thank you very much for your attention. And, I'll be around, and I hope I get a chance to talk to some of you. Thank you very much.